Professor Tobe, please.
Okay. Thank you very much for introduction. Hello, everyone. I'm Ryuta Tobe, an associate professor in the Laboratory of Microbiology at Tohoku University. At first, I'd like to thank the organizers for the opportunity to talk about our recent work. Actually, usually we focus on the pathogens, including in the infraction of influencing diseases of animals. So my talk is a little different from the purpose of this section. But anyway, I'd like to talk about something at this title. And this is the content of my talk. First, I will explain the food poisoning and its causes first, shortly, and then I will talk about antimicrobial peptide and pathogenic IP. It's one of the AMP we are interested in these days. And the last one is the establishment of the microbial production system of IP. First, food poisoning. It's called foodborne diseases or illness also. And the recent report from WHO, World Health Organization, they said an estimated 600 million, almost 1 in 10 people for ill after eating contaminated food, and for 20,000 die every year in the world. And 110 billion US dollars is lost each year in the productivity and medical expenses resulting from unsafe. Actually, unsafe food causes more than 200 diseases. And also, children under 5 years of age carry 40% of the foodborne disease burden, with 125,000 deaths every year. It's so sad. And combined with the spread of antimicrobial resistance, it's another serious problem by bacteria. So microbial problem become more serious in the world. So what is the main cause of the foodborne disease? Food poisoning is usually infectious or toxic in nature and cause bacteria, viruses, and parasites, and toxins and chemicals. The percentage depends on the country. Here is Japan, examined Japan 2021. And as you can see, in Japan, in the case of Japan, virus and bacteria account more than 90% of all of food poisoning. And more than half of them is bacteria, caused by bacteria. And in the case of virus food poisoning, it's caused by norovirus. And parasite also from anisakis. They are only one species. Only one kind of organisms. But in the case of bacterial food poisoning, it's caused by several kinds of species: Salmonella, Staphylococcus, E. coli, Clostridium, Bacillus, and Campylobacter. So it makes them control difficult. And generally, heat sterilization is recommended to remove these pathogenic bacteria, including virus. However, the Clostridium and Staphylococcus and Bacillus, they are known as heat-resistant bacteria, or they have heat-resistant toxins. So only heat treatment is not enough to remove the risk from them. Basically, bacteria can categorize two groups. One is gram-positive, and another one is gram-negative. Gram-positive bacteria have thick cell walls, consist of peptoglycan. And another gram-negative bacterium have thin cell wall and outer membrane. This heat-resistant bacteria is categorized into this group, gram-positive. Against the gram-positive bacteria, niacin is widely used as antibacterial additive, like additive in the food. So niacin is one of the antimicrobial peptide. Niacin is with 34 amino acids, like this. It has a special amino acid, lanthionine, and also it's produced by lactic acid bacteria, Lactococcus lactis. And niacin is effective against many gram-positive organisms, including the Clostridium and Staphylococcus and the Bacillus too. So niacin is used production of processed cheese, meat, and beverages to inhibit gram-positive pathogenic bacteria. However, niacin is unstable and inactive above the neutral pH. So its use as a food additive is so limited. Including in the niacin, AMP, antimicrobial peptides peptides are focused on the antibacterial drugs. AMP is part of the innate immune response found among all classes of life. And also generally between 12 and 15 amino acids. It's a small molecule. And its AMP is likely to cause resistance than conventional antibiotics, like penicillin or vancomycin or something. And several kinds of, a lot of kinds of AMPs reported. For example, from the several organisms. For example, defensin 2 and LL37 from human, and polyphenylmycin is, I forgot, horseshoe crab. So for more than, I think, 4,000 kinds of AMP has been reported in 2024. And its microbial mechanism of AMP is commonly bind to bacterial membrane, and then insertion and making a pore on the microbial membrane. And also, interestingly, recent studies show the AMP has another function other than the pore forming. For example, tannagin from insect is making pore, and also attack to the inactivation of NDM1. It's a drug degrading enzyme. And also human beta defensin 3, it makes pore and also inhibit biosynthesis of cell wall. So recently we found novel AMP from tick. It's a pathocalcin. We called it IP. IP is high antimicrobial activity against gram-positive bacteria, same as niacin. But it's more effective, like to gram-positive bacteria, S. aureus, bacillus, and Corynebacterium. On the other hand, it's not effective to gram-negative bacteria. And more important thing is IP is very effective to even drug-resistant bacteria like MRSA and VRSA. However, the mechanism of antibacterial actions is still unknown. So we try to clarify the mechanism of the IP, how to kill the cells. So we try to analyze the membrane permeability and also cell viability. Left one shows a cell membrane permeability. It means a membrane damage to the cells. We use S. aureus as a test strain. And red one shows the IP, and also black one shows the daptomycin. Daptomycin is also an AMP. It's already used clinically. So you can see the graph on the left side. The cell membrane permeability, the cell damage, increases gently compared to the daptomycin. And also does not show the immediate bacterial cells kill compared to daptomycin. Daptomycin is very quick, but IP is not so quick, it's slow. So we think IP has a different kind of mechanism from the daptomycin. Therefore, to elucidate the antibacterial activity of IP, we screened the IP high-sensitive mutant using a single gene deletion mutant library of S. aureus. It includes 2,000 strains. Each cell has a different kind of mutant, each genes. Using them, we screened the IP's high-sensitive mutant, 16 strains. And then we identified the genes, which genes is deleted in this mutant. The result shows, of course, we could identify the cell membrane stress response protein because it's IP also AMP, so it's bind to the membrane, cell membrane. In addition to that, cell wall stress response protein was identified. These results suggest this IP not only act on the cell membrane, but also cell wall or cell wall biosynthesis. Actually, and we need more, sorry, I forgot. Skip the one slide. What's that? Okay. Sorry. It's okay. So we need more experiment to clarify this mechanism, which we are still going on. And another project, we tried to the production of recombinant IP. We have two purposes. One is a further detailed analysis, like the action of mechanisms, and also we want to know the activity to other species, including the virus or other species, and also registration, resistance to the IP. And another one in the industrial application of IP, like niacin. For that, we need to know the stability and storages of IP, and also allergenic reactivity. And anyway, we need to make IP as cheap as possible, because we use chemically synthesized IP, but it is very expensive. You can see the 250,000 Japanese yen per grams, so expensive, so we cannot use so much. And for there is the following is microorganisms generally used for recombinant protein. producing recombinant protein. Bacillus subtilis is gram-positive, and the E. coli is gram-negative, and yeast is eukaryotic organisms. So, of course, we cannot use Bacillus because it's gram-positive, so it's sensitive to IP. And also, we tried to E. coli system, but in the case of E. coli, IP became the insoluble and inactive form. So we tried yeast system. The gene for IP is cloned and introduced into the Pichia bacillus yeast. And we got IP high expression strains. The strain incubated in the medium, and IP genes are induced by addition of methanol, because methanol is promotes gene expression. In this system, the produced IP is secreted out of the cells. So we check the antimicrobial activity in the supernatant, I mean the medium. As you can see, after induction, 0 day, 1 day, 2 days, 3 days, even 1 day, you can detect we observed antibacterial activity. But third day is much more, so we decide to use a third more induction. And to purify the recombinant IP, we use ion exchange columns. The medium applied into the columns and washed and eluted 2 kinds of buffers. And only addition 2, we use a buffer 2 is show the antibacterial activities. And then to check the purity of the protein, we apply the diffraction onto the SDS page. The flow through and force fraction and addition 1, 2, and chemically synthesized IP. As you can see, the recombinant IP was obtained with high purity. And then we try to check the stability of the recombinant IP. The left one shows treated with high temperature, 60, 70, 80, 90, 100, around 30 minutes. And the right one shows a pH change. It's 24-hour treatment. And lysine is control as a control. In heat treatment, more than 100 centigrades, and don't reduce the activity of IP and lysine. And no loss of IP activity was observed after storage and even high pH. On the other hand, lysine is a little decreased. And also, we try to check the toxicity of IP to mammalian cells. IP has almost no toxicity to microbial cells, mammalian cells, to epithelial cells and fibroblast cells. So we will try to analyze in vivo in the future. And it is a summary of this project, but I think it's not enough time to explain all of them, so I will just show it. I want to say thank the lab members, and this research is almost his work. So Shimoda, he got his PhD in this spring. And we need to appreciate the collaborator, Professor Nagagawa and Ito, Dr. Ito. Thank you very much.